Okay, hi everybody. This will be the lecture portion of Lab 7, the Gymnosperm lab. So, this lab is going to have three different portions, all of which will involve looking at pictures of different gymnosperms and either IDing them or just making observations about them and answering questions in the lab. So it's pretty similar to what we do in lab. It's just unfortunately we won't have any live tissue to look at and dissect. But hopefully we have put together a nice array of photos so you guys can become more familiar with the gymnosperms. Okay, so for the first part, we're going to look at branching and leaf structure, so the vegetative part of the plants, looking at how different gymnosperms deal with branching, axillary meristem growth, and types of leaves, organization of leaves, things like that, thinking about how the branching pattern will relate to the phyllotaxy of the main stem. And also with gymnosperms, since these tend to be long-lived and many of them living in a temperate climate, we have seasonal changes in growth patterns. So often this will involve long shoots and then short shoots. Bud scars will be present from when the bud opens, from the overwintering. And generally, the growth will be periodical in things like the pines and other gymnosperms like ginkgo that are temperate species. Okay, so let's practice doing this. Here we have a pine little tip of a branch. Starting from the bottom here, we can see that we have branch scars all around here. Then this is where we see bud scale scars. So in the past, this was two separate little buds overwintered, and then they broke open to produce this long branch. So if we follow this long branch, then we see another bud scale scar where we had a second year of a bud and then subsequent growth right here. So you can see right there. So let's zoom in on that. Okay, so it's a little deceptive the way the branching and the phyllotaxy is working in the pines. So when lateral determinate organs are born on a long shoot, they will have the identity of being a scale leaf. So we have to look closely to see these. They will fall off. They're just very small. And so here we see a scale leaf, and then what do we have in the axil of every lateral determinate organ? In this case, in the axil of our scale leaf, we have an axillary meristem, right? And what comes from an axillary meristem? A branch or a modified branch, right? So these are called fascicles. That's where we have a determinate short shoot that doesn't undergo any further growth than the leaf production, right? It stops growing and it never grows again. That's why I remember when we were in Harvard Forest, we were trying to follow back, see how old of leaves we could find on a pine branch, and we couldn't really go back that far because once that branch falls off, it will, in most cases, not grow again. Okay, and so these are just short reduced branches, and they have a whorl of leaves. So we have these suppressed internodes, and we get a whorl of these leaves. And we can also look at the leaf structure, right? These are very long and thin. They're a little bit flattened, and observations like that we can make about these species will help us learn how to identify them when we're out walking around. So let me move my little box. So pine scale leaves are arranged spirally. So see that? We can see that these are arranged spirally up this stem. And then the photosynthetic leaves that are born on the short shoots are born in fascicles. So it's interesting that the leaves on the long shoots, they have a different phyllotaxic pattern, and they also have a different identity. And then when we switch to the developmental program of the short shoot, the fascicle, the determinate branch, we can see that leaves are long, thin, photosynthetic, well-developed, and determinate. Okay. So we will wait for this part, and I will start to draw because the second part of activity 1 will involve a dichotomous key. So I just want to review quickly so we can all be aware. So here we go. Okay, so here we have 5 types of fruit. Despite my drawings, I will go through them. Banana, pear, apple. These are blueberries, and this is an orange. Okay, so if we did not know what these fruits were, we wanted to identify them, we could use a dichotomous key. So the way to use this is we're going to have different levels of organization. Each is indented, and we'll have a different bullet like thing. Like these are capital, these are lowercase, these are Roman numerals, so it can be numbers or it can be anything. But we're gonna pick one of our mystery taxa. So let's pick a pair. And we're gonna start from all the way left and then proceed right. So if we start all the way left, we see that we have A, round in shape, or B, non-round. So we'll say a pair is non-round, and then we'll proceed to the next subcategory in the one that we've selected. So yellow or green, it's a pair. So using these is a good way to organize lots of traits that can help you identify different groups. And so, we could go through for all of these. You could go through on your own if you want to pause it. Blueberries, you'll start from the left, rounded shape. Do they have a leathery or a thin peel? They have a thin peel. And then are they red or are they blue? They're blue, and that's a blueberry. So I hope this is helpful. We're just going to have a little dichotomous key in part 2 of activity 1 to try and identify some of the gymnosperm pictures. Okay. So then, activity 2, we will be going over reproductive features of male and female pine cone cross sections. And right, so I'm going to ignore this dissection step. Okay. So for the reproductive structures, I'm just remembering that the male cone is a simple strobilus, and we're going to go over that more, meaning that the sporophyll, the leaf, will bear directly the pollen sacs, microsporangia, and the female cone will have a sterile bract, that's our lateral determinate organ, and then our actual ovuliferous scale is a modified branch. And that's the difference between simple and compound strobilus. Okay, you can see in this picture here, this is a simple and this is a compound on the left. Okay, so for the simple strobilus, this is the male, that's the microsporangiate strobilus. Sporangia are born on sporophylls. They're born on the bottom of sporophylls. This is different from in the female cone where they're born on the top, what we call the adaxial side. And in the simple strobilus, the microsporangia are born on the abaxial side of the sporophyll. And the male cone will be reminiscent of our lycophyte cones, the lycophyte strobili. This is a selaginella. You can tell because we have heterospore here. But if we were looking at just a lycopodium, we would have homosporous, and it would look quite similar, as it's just they're both simple cones. And fundamental difference will be that this pine cone is producing pollen, whereas this is producing microgametophytes, which will be dispersed and then will germinate separately and then produce sperm, not pollen. Okay, so yeah, we're gonna look at some prepared slides. Here's an image of this microsporangiate cone. You can see you have all of these sporophylls here coming off, spirally arranged on a main axis, and the sporangia are going to be born on the undersides of these leaves. And pollen, remember this is our big innovation with the seed plants. It looks like this, kind of Mickey Mouse ear shape when you view it in cross section, and you have these big, well, first of all, in the main body of the pollen, you have two cells. You have a tube cell and a generative cell. Now the tube cell is going to grow, it's going to move down the pollen tube as it lengthens. It kind of directs the growth of the pollen tube. And then the generative cell will split to form 2 sperm, which will then go ahead and fertilize the eggs. Fertilization can happen over a year after pollination occurs. So from when this sperm right here will land on the pollination drop, and from the time this generative cell will divide to produce sperm and the sperm will fertilize with the archegonia is a year. It's a long time. Compare this to the angiosperms, where you will go from pollination to seed production within 1 year. Also, we have these big air sacs. Those are called air sacs. And initially, these were thought to help the pollen fly. What we think that these do is actually to break the surface tension of the water in the pollination droplet. Okay. Okay, so now I will we'll start with the male cone. Let's think about what's going on inside of there. Okay. So we start with a sporophyll. It bears 2 pollen sacs. We'll just view it in side view. It's a pollen sac. That's our microsporangium. Okay. Okay, so inside this microsporangium, what are we going to do? We're going to undergo meiosis to produce haploid spores. We've undergone meiosis, and now we have a microsporangium packed full of spores. What do these individual spores do? So remember that the male, in contrast to the female, will have many microspores, whereas the female will just have 1 microspore. So microspores, okay. And what do microspores do? They grow by mitosis to form our microgametophytes. What is that? That's our pollen. So if we blew one of these up, it will be our 2 air sacs and our 2 nuclei, and that is our whole microgametophyte, which will then go to fertilize an egg. So let's compare this with the female cone. Let's just see here. So here is a female cone, which is all the way expanded. This is after it has released almost all of its seeds. You can maybe see a wing of a seed right there. But the pollen cone, when it gets fertilized, is much smaller, more delicate when it gets pollinated. And then it will grow. I think we have a picture. Let's skip. Okay. Yeah, so over here we have, this is where pollination will occur. So this is ready to receive pollen. It's secreting a pollination drop. Once it's pollinated, while we wait for the pollen tube to grow all the way down, finally fertilize the egg, it will grow into something like this. So it's kind of fleshy. We start to see, oh, and I should note right here that this is a compound strobilus, remember? So we're going to bring ourselves back to this mindset. So we have tiny, so here's one, tiny sterile bracts. This is not, I think they should be pointing like to this or something. These are in the wrong spots, I think. But so here we have a sterile bract, here we have a sterile bract, and then this with our ovule is our ovuliferous scale, which is our modified branch. So if we look at this, our cone ready to receive pollen, our megasporangiate cone ready to receive pollen, we see these leaves, and then we see the subtending ovuliferous scale. So we can see these scales a lot better when the cone is fresh and small. And then these ovuliferous scales will grow just basically to totally block the scale. They just outgrow it. And then once we get fertilization and we get seed set, it will harden, it will desiccate, and then the scales open by flexion and the seeds can disperse. So let's just go over the female cone, because there's kind of a lot going on there. I just want to remind everybody we can just go through it. So there's many layers to the female cone scale. So first we get the scale right with our ovule. This is our ovuliferous scale. Okay. It has a subtending sterile bract. I'll just do SB. And remember what this is. This is a megasporangia cone. So we're going to have, so here's our integument, because what is an ovule? It's an integumented indehiscent megasporangium. So here's our integument. And then inside of here, you're going to get your megasporangium. Okay. So let's make this green. Okay. So there's our megasporangium. So what happens inside the megasporangium? It undergoes meiosis, right, to produce a megaspore. And so what we will actually get within the megasporangium is a megaspore mother cell. Only compare this to the male, where we have a bunch of microspores. In the female, we're only going to get, well, we're going to have one microspore, but we're going to have only one meiosis event, only one megaspore mother cell, which will divide by meiosis to produce 4 haploid microspores. Remind you that this is all diploid tissue. And then 3 of those cells will die, right? So we're gonna, after we undergo meiosis and we undergo death of 3 out of 4 cells from meiosis, then we will have our integument, we have our megasporangium tissue, and we have a megaspore. Okay, so now we have megaspore inside of diploid tissue. The megaspore is haploid. And what is a megaspore going to do? It's going to undergo mitosis, right? To make a megagametophyte. So we have our our diploid nucellus tissue here. So our megaspore is gonna, let's make it a different color. Start making this blue. So that's our haploid. So this is gonna divide, produce our megagametophyte, right? So now we have this 2N tissue. This is now haploid tissue. And what is the megagametophyte going to do? It's going to produce eggs. Mitosis, right? Since we're haploid. And so what will happen is it will produce archegonium. And per ovule, you will get 2 archegonia. So these will produce eggs via mitosis. I'll label these our archegonia. This is our megagametophyte. Right. Okay, so now I'm just gonna zoom in. I'm gonna draw it. I'm gonna draw just like this part, okay? So now what do we have? We have an integument. We have nucellus tissue, which has been pushed out by our megagametophyte. We have 2 haploid eggs. We'll just draw the egg like that. So let's color this in. We'll color this in. Okay. So this our megasporangium. We call this nucellus. So we have a layer of nucellus. Then we have a layer of our haploid gametophyte. And then these eggs will become fertilized. We'll make them purple. So fertilization happens. And then we will get what? Embryo. One of these will die. So we have mitosis of zygote. And then we will get an embryo. So all together, once we have a seed, right? We have the integument of the the ovule. We have a thin layer of nucellar tissue, which inside of that is our megagametophyte. That's our nutritive tissue. And then we will have haploid megagametophyte tissue, inside of which you will have a embryo, diploid embryo, right? Okay, so hopefully this will be helpful when we are looking at cross sections of the female ovuliferous scale. So if we look at this zoomed in version here, what do we have? We have our integument, that's like part of the scale. Here's our micropyle, that's our opening root. So we'll have a pollination drop secreted in here. Then we have this is our uh, reminiscent megasporangiate tissue. So our diploid megasporangiate tissue. Within that, we have haploid megagametophyte, nutritive tissue, and then here is an archegonium within which an egg will develop and we will have an embryo. So all of this tissue is involved in formation of the seed. And as this megagametophyte grows, as everything grows, the megasporangia tissue just kind of gets pushed out. And so we basically end up with a seed coat and then a haploid megagametophyte layer within which there will be a developing embryo. So here's a nice cone. You can see right here we have these sterile bracts. There's the cross section right there. In this, they're much bigger. And then these non-spiky ones are our ovuliferous scale. So our ovule will be down within there. And remember that on each ovuliferous scale, you normally have 2 ovules. They're symmetrical. It's worth appreciating the incredible diversity of cones and reproductive structures in gymnosperms. They're so beautiful. I didn't really appreciate them enough until recently. Thinking about all these modifications that have been made, convergences with flowering plants in terms of making kind of like a fruit-like structure by sometimes having a fleshy cone or sometimes having a fleshy aril, the base of the ovule. And yeah, all the beauty in the different stages of cone development. Like when they are young, they're so pink and cute. Yeah, and there are some more pictures. These are, looks to me like these are all male cones, all microsporangiate cones. So you can see these are Taxaceae cones. All of these kind of looking similar. And all of these will be simple cones and will be wind dispersing their pollen. So yeah, lastly, we will just draw the seed plant life cycle. I will do that on my phone, and then that should be it. So good luck with the lab. Okay, so right now we're going to go over the generalized life cycle of gymnosperms. This will apply to any gymnosperm, and we're going to talk about that as we go. So, okay. So we can start with our diploid individual. Has a stem, has leaves, and this is a 2N. So let's say this is a monoecious species. It's going to produce megasporangia and microsporangia cone via mitosis. Let's start with the megasporangia cone. So starting with the megasporangiate strobilus, this is going to produce an ovule. Remember that an ovule is an integumented indehiscent megasporangium. So what will we have? Here's our integument. That's our diploid sporophyte tissue. And on that, we will get megasporangium here. And within our microstrobilus, we're going to have on one of the sporophylls And on that, we will have a microsporangia, aka pollen sac. Okay, so let's take stock here. We have a megasporangiate strobilus, microsporangiate strobilus, on which we're getting sporophylls, which on the megasporangiate cone will produce an ovule, and on the microsporangiate cone will be a pollen sac. So within our ovule, we still have our integument on the outside, megasporangium on the inside. Now, what happens inside of this megasporangium? Obviously, this is still diploid tissue. We produce all of this by mitosis, but our goal in making a sporangium is what? To produce spores. And to produce spores from a diploid structure, we need to undergo meiosis. We'll undergo meiosis. We just quickly switch back to the microsporangia cone. It's the same situation, right? We have a diploid microsporophyll, diploid pollen sac. Our goal is to produce haploid spores so that we can go on to produce pollen, our microgametophyte. So we need to undergo meiosis within here. Now, there's a couple important differences between the microsporangiate cone and the megasporangiate cone. So in the microsporangium, we will get this pollen sac will fill with hundreds or thousands of microspore mother cells. And all of those cells are going to undergo meiosis. So we have hundreds or thousands of meiosis events going on within here to produce hundreds or thousands of microspores, which can then germinate to form pollen. That's many. In contrast, what we have right now in our ovule is a megasporangium. But what's actually going to happen within that megasporangium is we will get a single megaspore mother cell. I will note that these are diploid, mini microspore mother cell, which then undergo meiosis and produce these haploid microspores. So now in our female, of course, this megaspore mother cell is going to undergo meiosis. This is the only cell in the megasporangium that will undergo meiosis, and we'll end up with four products from that meiosis event, 3 of which will die. So we will end up with a single megaspore. There's our haploid megaspore. These haploid megaspores in both our microsporangium and megasporangium are going to undergo mitosis to form our gametophytes. Okay, so at this point, our micro and megaspores are going to divide by mitosis. Okay. And so what do we get when our microspores undergo mitosis? We get our microgametophyte, and that's our pollen, right? Okay, and our haploid megaspore is going to divide to produce our megagametophyte, right? And of course, this haploid megagametophyte is going to produce archegonia via mitosis. So in our female, our megagametophyte can be thousands of cells. That's our nutritive tissue. Our microgametophyte is only 2 cells at maturity. And we went over earlier in the lecture the structure of pollen. So we can just be thinking about how these heterosporous individuals have evolved such different programs based on whether they are making eggs or sperm. So let's step back and just look at what we have now. We've gone from a diploid mature individual gymnosperm to having sperm, and let's just go ahead and make this an egg already. We know our archegonia are going to produce eggs, so these different parts will vary. Like when we talked about the pine scale, you remember we were talking about, in the case of the female, a woody pine cone. So we've got megastrobilus with many, many ovules inside, spirally arranged. But if we think about other gymnosperms, that condition might vary. Like in the ginkgo, we just have, in the ginkgo, we'll have single ovules, two ovules born on a stalk. Here's our pollination drop. But regardless, within that ovule, we'll have two end tissue on the outside with a megasporangium on the inside. And essentially, the things that happen within the sporangium will be similar. So also what we can notice about our ovule is that it will have a micropyle. Let's do the MP. That's the gap in the integument that allows our sperm to come fertilize the egg. Okay, so eventually we will get release of pollen. Our megagametophyte here will secrete a pollination drop at the micropylar end, through which the sperm will enter the megagametophyte. They will land, break the surface tension of the water, likely with the air sacs of the pollen, and then the pollen tube will grow down into the megagametophyte to fertilize the archegonia. Okay, so upon fertilization, we will get an embryo. So let's just refresh ourselves with what we have now inside of a fertilized ovule. We will have a parental sporophyte tissue, that's our integument, followed by our megasporangia tissue. That is also called our nucellus. We have our haploid megagametophyte. Okay, so now we have our fertilized egg, our embryo, and what are we going to make in every single gymnosperm at a seed? Okay, so this will lead us lastly to our seed. So thinking about the parts of the seed. So with our fertilized embryo, we have a layer of, this is our integument, our light green integument. It's our diploid parental tissue, inside of which there is a thin layer of nucellus. And this nucellus, remember that our gametophyte is entirely nutritionally dependent on our sporophyte. And so the megasporangium tissue will help to nurture the growing megagametophyte, which in turn will later nurture the growing seedling and the embryo itself. So in our mature seed, we get drying and hardening of the seed coat. So we have a thick seed coat here derived from our integument. Now we will have a tiny, sometimes visible layer of nucellus in here, but for the most part it will be completely obscured by our mature megagametophyte. So let's just put our megagametophyte in there. That's our haploid nutritive tissue. And then within that, we will get our embryo. So this is like our pine nut. Remember, we ate the pine nut, the seed coat had already been removed. And so basically what we were eating is just this light blue megagametophyte with the embedded embryo. I know some people were able to find embryos. And the seed will be varied between gymnosperms. In the pines, the integument forms a wing. In the ginkgo, it will actually be a fleshy seed coat to promote dispersal, so a lot of convergences with fruits and angiosperms. So thinking about this central life cycle for all the gymnosperms and how it is varied between them will be helpful, and then we can really come to understand the gymnosperms. And so the last step of this will just be germination. And we will have our first leaves here and our radicle. And then this will grow by mitosis into a mature individual, and the whole cycle will start again. So I hope this was a helpful review, and good luck with the lab. Bye.